In this video, we'll start talking about the blood supply to the kidneys. Now, at rest, a large amount of the cardiac output is dedicated to go into the kidneys. So the kidneys receive about about 1/4 of the total cardiac output. So that comes out to about 1,200 milliliters of blood per minute. So they're highly vascularized, and it makes sense because the kidneys are about filtering the blood and doing all those homeostatic controls based on the blood. So the renal arteries are fairly large arteries, and they branch into smaller vessels, and they work their way through the renal sinus on their way up to the renal columns and the cortex. So really the path is from a renal artery to a segmental artery to an interlobular artery to an accurate to a interlobular versus interlobular. I know it sounds the same but slightly different. And these are also known as the cortical radiate arteries. And then this leads into a group of capillaries. Now, when we're talking about the capillary bed associated with the kidneys, they're not your standard capillary bed system. The arterioles both feed and drain the capillaries. So, in many ways, this is quite different than what we've seen before. Now, each of these interlobular arteries leads to what we call an afferent or an afferent, arteriole. And this leads into the glomerulus, which is sort of like this ball of capillaries. And coming out of the glomerulus, we have the efferent. So going in, heading in, we have the afferent. That becomes a ball of capillaries. And then coming out, we have the efferent. So the path of the blood is from the afferent into the glomerulus, where certain things will leave the capillary bed and enter the nephron, and any of the blood that is not filtered out into the nephron leaves by the efferent. And then also we have the efferent that carries on as what we call the peritubular capillaries. So we come down here, this is an arteriole, it enters into a capillary bed. So that was the afferent going in, the glomerulus. Coming out of the glomerulus is the efferent, and then the efferent, as I said, carries on, and it then becomes something called the peritubular capillaries. Now this is going to give you a little hint. Peritubular means they're near the tube, and they're going to act as capillaries as we go. So it's basically the afferent into the glomerulus, into the efferent, into the peritubular capillaries. And then from there, eventually, of course, the venous blood has to be drained out. And we go from the interlobular to the accurate, to the interlobular, to the renal and vein, and then that exits the kidney and heads to the vena cava. And we can see some of that represented here. Now, we can see some of the structures we've talked about before. We have the pyramids, and in between the pyramids are the columns. So here we have the renal artery heading into the segmental arteries, because it goes to different segments. And then heading into each lobe are the interlobular arteries. And you can see how it arches up here. And then when we zoom in, we can have a look at the diagram over here. We can see that the artery, that's the artery that's arcing over, sends off branches. And here you can see, if we watch, we can see that here an afferent, starting with the letter A, enters that ball of capillaries, which is the glomerulus, and coming out is the efferent. Now the efferent, as we said, in most of the nephrons, will head off and form this structure here, which is the peritubular capillaries. And you can see why, is that they really are associated with the tubes that we have. Okay, so that's a bit about the blood flow. Now, this is going to make more sense once we start talking about the functioning of how the glomerulus works. But we should start off by at least having the basic understanding of the blood vessels involved. So So let's start talking a bit about the structure of the nephron, just to get some of the real basics down, and before we head off into the detailed structure and function. So we have the renal corpuscle, and what does it do? It's responsible for filtering blood. Now, this renal corpuscle is actually composed of two parts. One part is the glomerulus, and this is a group of capillaries, but it's not your regular capillaries. These are fenestrated. Remember what that means is that these are very leaky, permeable capillaries. And then surrounding the glomerulus, so surrounding the capillaries, so we have the ball of capillaries, surrounding the ball of capillaries, we have Bowman's capsule, also known as, well, most people now call it the glomerular capsule. Bowman's is the other way, the old name for it. This is a double-layered outer container of epithelial tissue that has both an outer parietal and an inner visceral layer. And as we look here, we can see the basic structure of a nephron, that we have the afferent going in, the efferent coming out. We have the glomerulus, and surrounding the glomerulus is this double-walled structure, which makes up the capsule. And then from there, you can see the tube starts, and the proximal convoluted tubule is the first, and the name makes sense. Proximal means it's near the start, right? Convoluted because it's very convoluted, and it is a tube, so there we go. So we have the proximal convoluted tubule. That eventually leads into this loop that dips down. Now it depends which type of nephron we're looking at. Some nephrons barely go down to the middle, and some go quite deep. Their loops go quite deep. But we can see that the loop goes down into the area of the medulla and comes back up. So that is the loop of the nephron. And then the third part is the distal convoluted tubule. Now, distal convoluted tubule dumps its contents into this duct, and this duct can filtrate from a number of nephrons. And since it collects and it's a duct, we're going to call it the collecting duct. So we have the proximal convoluted tubule, distal convoluted tubule, and the loop of the nephron, or the main parts of the nephron. Okay, so let's just talk a bit about the renal corpuscle. So we have the glomerular capsule, and this is that globe-like part. It's an extension of the renal tubule made of simple squamous epithelium. And we have a visceral layer, which is modified epithelial cells. And we give them a special name, podocytes. Now, podo refers to foot, like podiatrist, that type of thing. So these are little foot processes, and what they do is they surround the capillaries and they form filtration slips. Later on, we're going to see that even though these are fenestrated capillaries, only certain things can come and go, or I should say go. Nothing really comes in. Things go from the blood into the filtrate. And then we have the capsular space, which is the area between the parietal and the visceral. Now, as I said, we have these podocytes and fenestrated glomerular capillaries, and this forms a very special membrane that really controls what can leave the blood and start to enter the actual nephron. So let's have a look. Here we have the afferent, which brings blood in. Of course, the efferent is what takes blood out, right? Efferent versus afferent. And then here are the capillaries, and you can see it forms this sort of ball of capillaries. And when we're looking at the glomerular capsule, out here is the parietal layer. That's the parietal layer. And then we have the inner visceral layer. Now remember, the visceral layer can form these podocytes, and you see how they sort of surround here. And they form something called filtration slips, because you can see there are slits. You can see there's little areas between the interdigitated, almost fingers of these podocytes. Now the space between the parietal layer and the visceral, so the parietal layer and the visceral layer, this space is the capsular space. So anything that enters this capillary bed and has the ability to filter out of the capillaries through these fenestrated capillaries enters the capsular space. And that material will then enter the proximal convoluted tubule and start to work its way through the nephron. And up here we can see an electron diagram or a scanning electron micrograph. And you can see these podocytes and how they sort of interdigitate. And we have those little filtration slits that are there. And this is one, not the only factor, but this is one of the factors that controls what's going to stay in the blood here and what can exit the blood. As you can imagine, red blood cells don't exit. White blood cells don't exit. Proteins, unless you're very small and very, very few, even if they're small, will exit. So hardly any proteins will actually exit here. But things like sugar can, glucose, electrolytes, water, etc. Some hormones, if they're not bound onto a protein, may end up in the filtrate. Right? Okay, so let's leave this video there, and the next one will come back and we'll start talking about the actual microanatomy.